Drop a like and do share. Leave your comments and do not forget to subscribe for more videos.
Hello students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is 5th section of the chapter Cell, the basic unit of life. In this section of the video, we will talk about cilia and flagella, that is a cell organelle which is present in eukaryote cell students. So cilia and flagella, they are hair-like outgrowths of the cell membrane students. Cilia, whose singular form is cilium, and flagella, whose singular form is known as flagellum. They are hair-like outgrowths of cell membrane. Okay? Cilia, they are the small structure which work like oars that causes movement of either the cell or surrounding fluid. Flagella, they are comparatively longer and they are responsible for cell movement. The prokaryotic bacteria, they also possess flagella, but these are structurally different from that of the eukaryotic flagella students. The electron microscope study of cilia and flagella, it shows that they are covered with plasma membrane. Okay, see, this is the plasma membrane which is present on outside of the cilia and flagella. Okay, their core is called as exoneme. So this is the core of a cilia which is known as exoneme. It possesses a number of microtubules. See, these are the number of microtubules which are present in the cilia. I have already taught you about microtubules, microfilaments, in fact entire cytoskeleton in my previous section of the presentation. So for better understanding, please refer to that. Okay. So exoneme, it contains many or several numbers of microtubules that runs parallel to the long axis. See, these all are running parallel to each other, right? The exoneme, they usually has 9 pairs of doublets, okay, of radially arranged peripheral microtubules and a pair of centrally located microtubules. Okay. So such an arrangement of exonemal microtubule is referred as 9+2 array. Now see, this is the arrangement. This is centrally located 2, or you can say a pair which is present in the inside of the microtubule. So this is your central microtubule. So they are 2 in nature. So 2, and these are 9 in nature. 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9+2 arrangement is seen in cilia and flagella. Okay, because their core is known as exoneme. It has many microtubules that run parallel to the long axis, and exoneme has 9 pairs of doublets. See, this is doublet and these are 9 in nature, and centrally located is central microtubule. So 9+2 array is seen in cilia and flagella. And please note that these central tubules, these two are connected by bridge. See, this is the bridge and is enclosed by a central sheet. See, this red one is central sheet, okay, which is connected to one of the tubules of each peripheral doublet by radial spoke. See, this is radial spoke. Okay. So central sheet, it is connected to one of the tubules of each peripheral doublet by radial spoke. So this is the connecting link between these doublet and central spoke. Thus there are 9 radial spokes, and the peripheral doublets, see, these are the peripheral doublets. Okay, see, these are the peripheral microtubules doublets. They are also interconnected by linkers. See, these are the linkers. This one are linkers. Both the cilia and flagella, they emerge from centriole-like structure which we call it as basal body. Okay. So the central tubules, they are connected by bridges as enclosed by central sheet, as I just taught you, and it is connected to one of the tubules of each peripheral doublet by radial spokes. Thus there are 9 radial spokes. See, these are called as radial spokes. So don't get confused with radial spokes. The peripheral doublets, they are also interconnected by linkers. Okay, see this green one is linker. Cilium and flagellum, they emerge from centriole-like structure, which we call it as basal body. Okay, so this was about cilium and flagellum. Now comes centrosome and centriole, students. What is centrosome, students? It is present only in animal cell and not in plant cell. Okay, so it is a unique characteristic which is present in animal cell. So centrosome is an organelle that contains two non-membrane bound cylindrical structure, which we call it as centriole. So this is centriole. Okay, it is an organelle which usually contains two cylindrical structure. As you can see, this is one cylindrical structure and this is another cylindrical structure. So it contains two cylindrical non-membrane bound structure, which we call it as centriole, and they are surrounded by amorphous pericentriolar material. Okay, the centriole, it lie perpendicular to each other. Okay, this way 90 degree. One is this and other is, and it always orient in the perpendicular direction. They are made up of 9 evenly spaced peripheral fibrils of tubulins. See, these are 9 spaced peripheral fibrils of tubulin, which is this centriole made up of. Okay, each of the peripheral fibril is triplet. The adjacent triplet, they are also linked. Okay, now the central part of the centriole is also proteinaceous, and it is called the hub, which is connected with tubules of peripheral triplets by radial spokes, which is made up of protein. See, this is your centriole, which is arranged in the right angle direction, that is perpendicular to each other they are arranged. This centriole is made up of microtubules. Okay, so the central part of the centriole, see, this is the central part of the centriole. It is also very proteinaceous and called the hub. This central part of the centriole is called as hub, which is connected with tubules of the peripheral triplet by radial spoke. Okay, which is made up of protein. The centriole, it forms the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. So centriole, it plays a very crucial role in cell division, particularly in animal cell and not in the plant cell, because it is a unique characteristic of animal cell. Okay, so centriole, it forms a basal body of cilia or flagella and spindle fiber that give rise to spindle apparatus during cell division in animal cell. So this was all about centrosome, centriole and cilia and flagella. In my next section of the presentation, I'll be teaching you about the nucleus and microbodies. So till then, stay tuned and keep watching Edupedia word videos. Thank you.